Hi, everyone, and welcome to the nervous system. Okay, so we're going to start here with a flow chart that summarizes the different categories of the nervous system, its subcategories, and so on and so forth. So, first up here, CNS, central nervous system. So, the central nervous system includes the brain and spinal cord. The CNS is responsible for controlling everything. It's the decision maker, it's passing those decisions along, and it's the control center of our body. So that's the CNS, brain, spinal cord, integrative, and control center. We also have the PNS, the peripheral nervous system. PNS. So that's the cranial nerves and spinal nerves. That's the other half of the whole. So central nervous system, brain, spinal cord, center of the body. Peripheral nervous system, PNS, periphery of the body. The PNS contains the moving parts of the nervous system. So, like I just said, the cranial nerves and the spinal nerves. Cranial nerves come out of the brain, and spinal nerves come out of the spinal cord. PNS is in charge of the communication between the central nervous system and the rest of the body. So, the PNS, the peripheral nervous system, it communicates from all the receptors in the body, the pain receptors, temperature receptors, pressure receptors, chemoreceptors. The PNS takes the data from the receptors and sends it to the central nervous system. So then the central nervous system picks up that data, it reads it, it makes a decision, and it sends back instructions to the PNS, and that way the PNS can carry out the orders given to perform whatever function is necessary to perform. An example would be if you touch a hot tea kettle with the side of your finger, the temperature receptors, the pain receptors, they would all kick in. So that data gets picked up by the PNS, moved through the spinal nerves into the spinal cord, up to the brain, and then the brain decides, oh, wait, don't want this, move that finger. And then the brain sends a directive to the biceps brachii muscle to contract, to pull your hand away from that hot tea kettle. The PNS has different categories. So, moving away from that example, peripheral nervous system breaks down into a few that break down into different ones that break down even further. Okay, so first, let's talk about the two divisions that branch off of the peripheral nervous system. So, before I do get into those, because we have sensory afferent and motor efferent, let's talk about what afferent and efferent means, because you're going to see it a lot in the rest of the semester of Anatomy 1, and then you'll see it a lot in Anatomy 2. So, afferent, efferent are going to come up a lot, A&P 1 and A&P 2, so learn them now. So, afferent, that means arrival. Efferent means exit. So, A, arrival, efferent, exit. If you remember that, then you're going to be pretty set, and you'll have to remember it forever, so you might as well learn it now throughout your anatomy career. So, again, afferent, arrival, efferent, exit. Again, if you remember that, you're going to be set for the rest of the year. Anyways, let's talk about these two divisions that are branching off of the peripheral nervous system. So the sensory division, the afferent division of the PNS, it picks up info and sends it to the brain. There, the brain can pick it up, make a decision, and then it'll get sent back out. Where does it get sent back out? It gets sent back out through the motor, the efferent division. So afferent arrives at the brain makes a decision, sends it through the efferent. So that decision is exiting the brain, right? So that's our motor efferent division. So the motor division, or the movement division, this division fires the motor fibers and is involved in functions that make you create a change or move whatever you need to do, that your brain just sent that information along. So in the hot tea kettle example, the motor division was that biceps brachii muscle activating so that your finger wasn't touching that hot tea kettle anymore, right? So the motor division conducts impulses from the central nervous system to effectors, muscles, and glands. Okay, so the efferent division, the motor division, actually breaks down into two other ones. We have the somatic nervous system, that's voluntary, and we have the autonomic nervous system, that's involuntary. So soma means body. Learn that now. It means body everywhere. You're going to hear about it in a different scenario here. You're going to hear about it next semester in A&P2. You're going to hear about it throughout your biology career if you go that direction. But soma means body. So somatic nervous system is our voluntary nervous system, conducts impulses from the central nervous system to the skeletal muscles. Autonomic is involuntary. In the somatic nervous system, so somatic, motor, voluntary, CNS to skeletal muscle, impulses are being sent from the brain spinal cord to skeletal muscles, and that leads to voluntary movement. So moving your body voluntarily. Sorry. Had to get a drink of water. Okay, so again, somatic, voluntary movement, information's being sent from the central nervous system to the skeletal muscles, and you're moving those skeletal muscles voluntarily. Now, on the other hand, autonomic nervous system sounds like automatic, right? Well, it's not automatic, it's autonomic, so make sure you don't mistake the spelling there. Autonomic nervous system. So this is involuntary. So think about things that happen automatically. They happen by themselves. You're not actively doing anything. So this one conducts impulses from the central nervous system, so that brain spinal cord, to muscles and glands. But when we're talking muscles here, we're talking cardiac muscle, smooth muscle, and glands. We're not talking skeletal muscle. Skeletal muscle was involved in voluntary, somatic, motor, so somatic nervous system. Autonomic is involuntary. We're talking cardiac muscle, smooth muscle, and glands. So you don't tell your heart to beat, right? You don't tell your vessels to constrict or dilate. Breathing, blinking. You can control them, yes, technically, if you start to actively think about it. But in essence, those are both involuntary because you got to blink to keep your eyes moist and you got to breathe to stay alive, right? So that's autonomic nervous system. The ANS, so the autonomic nervous system, further breaks down into two other divisions, the sympathetic and the parasympathetic division. Sympathetic is associated with fight or flight. So sympathetic mobilizes the body systems when you sense danger. It makes your senses hyper acute. So think evolutionarily. The sympathetic nervous system is going to kick in when you start sensing danger, like being chased by an animal. So when the sympathetic division of the autonomic nervous system kicks in, your eyes are going to dilate so it can sharpen your focus in low light. Your hearing is going to become more sensitive. Your muscles are ready to kick in, whether to run or to fight. All of your senses are going to be hyper acute. You're going to be hyper aware. That is your sympathetic division of your autonomic nervous system, which is part of the motor efferent division of the peripheral nervous system, right? So know this chart. You're going to be asked to track all of these, like I just tracked the sympathetic nervous system. So sympathetic, fight or flight. Parasympathetic is the opposite. Parasympathetic, we use the phrase rest and digest. So with parasympathetic, your heart rate's slowing down, there's less blood flow to the extremities, your body's conserving energy everywhere except for digestion. This is when your body is focusing on digestion. So it promotes housekeeping functions during rest. So the parasympathetic division, it's basically most active while you're sleeping. It's low and slow, right? Like I just said, except for with the digestive system. And that's why actually you have to go to the bathroom when you wake up, because your digestive system has just worked to rest and digest. Or, well, not to rest, right? Parasympathetic is rest and digest. So everything's resting, hanging out, calming down, except for your digestive system. But your digestive system is digesting. So you wake up, go to the bathroom, because your digestive system was just working while you were sleeping. And then we'll talk more about the autonomic nervous system in future chapters. All right, so our two major divisions again are our central nervous system and our peripheral nervous system. So in blue, that's our CNS, our brain and spinal cord. It's in the center of our body. In yellow in this image, all of those little wispies coming off the brain spinal cord, that's our peripheral nervous system. So those are our cranial and spinal nerves. Peripheral are on the periphery, so edges of the body. Let's see. I think that's it there. You have to know this. So make some flashcards. Make some flashcards for this entire chart because you're going to have to know it. Draw out this chart and fill in the blanks because you're going to have to know it. And you're going to have to know how everything breaks down. And the categories, subcategories, etc., etc. All right, so breaking it down further. So reiterating slide 2, that's what these next few slides are going to be. And we're going to break the peripheral nervous system down to two divisions, like I just talked about, afferent and efferent. Afferent was sensory, efferent is motor. So afferent is sensory from the periphery to the CNS. Efferent is the motor from the CNS to the muscles and glands. In this picture, something is affecting the skin. Nerves are picking that up and sending it via the PNS to the spinal cord onward to the brain. Again, spinal cord, brain is CNS. So then the CNS, the brain, sends a signal back down through the spinal cord to whatever muscle or gland needs to work to make whatever change is necessary. So sensory up to the CNS, and then motor, efferent, down. Afferent is going to arrive at the brain. Efferent is going to exit the brain and make whatever change is necessary. Going on down the chart, like I just said, breaking afferent sensory division down further to its somatic and visceral categories. So our somatic sensory fibers are from the skin, skeletal muscles, and joints to the central nervous system. And then our visceral sensory fibers, visceral, easy enough. Visceral, when we talk visceral, we're talking organs. So this is from visceral organs to the CNS. Again, soma means body, so muscles and joints are going to be the support structures of the body. Visceral is always going to refer to guts, internal organs. And then, again, afferent is to brain and spinal cord. Efferent is away from brain and spinal cord. So these afferent divisions are going to the CNS. And then all of these have pain receptors, temperature receptors, stretch receptors, chemoreceptors, all the receptors, and they're sending information to the brain and spinal cord. Again, soma means body. And then visceral, we're talking organs. So somatic sensory fibers from the skin, skeletal muscle, and joints to the CNS. Visceral sensory fibers from visceral organs to the CNS. And then the efferent. So going back up here to slide 2. Now we're on the efferent divisions. Break down into the somatic nervous system and the autonomic nervous system. So this is information exiting the central nervous system, the brain, the spinal cord, going where it needs to go. This is the counterpart to the efferent, or to the afferent division, right? So here we have the somatic nervous system and the autonomic nervous system. Like I said earlier, somatic is voluntary. So motor from CNS to skeletal muscle. So contract that biceps brachii muscle to get your hand away from that hot tea kettle that your finger touched. This happens really quickly, like a reflex, but it's still voluntary because you can choose to leave it there. So some people like pain, and there are several psychological examples that we could delve into, but we won't in this class. There are so many different, there's a reason that psychology is its own major, right? There are so many different scenarios, et cetera, et cetera. We're just going to talk about anatomy, physiology. Again, some people that like pain are not going to jerk their hand away, right? That's where somatic nervous system is voluntary, right? So even though that tea kettle is hot, maybe somebody's going to leave their hand on it. I mean, that's a bad example for something like that, but still, it just goes to tell you that somatic nervous system is voluntary. Autonomic is not. So the second efferent motor division is the involuntary one. The autonomic one sounds like automatic. You are not in control. So this is from the central nervous system to smooth muscle, cardiac muscle, and glands. So in this picture, we have a piece of intestine and a sensory neuron taking information up to the CNS, to the brain spinal cord in green, and then back down and out to the intestine with the blue there to indicate that. And that way the intestine can do whatever it needs to do, like maybe stretch or squeeze to help move waste along the intestinal pathway. But this is stuff that you're not in control of voluntarily. This is just what happens, what needs to happen involuntarily. Okay. All right, let's break it down even more. The peripheral nervous system, efferent autonomic nervous system, has two divisions: sympathetic and parasympathetic. Right now, just know that sympathetic is fight or flight and parasympathetic is rest and digest, and we will talk more about it. You can read through this, but we'll spend an entire chapter on just the autonomic nervous system, the ANS, sympathetic and parasympathetic. So this is a practice chart. Practice, 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 because you will be asked and expected to know this for the rest of the semester. Don't just learn it for this quiz. Practice it. Incorporate this into memory, which we'll talk about. Memory at the end of this chapter. But yeah, I could ask a question like, which of the following is not a branch or division of the PNS, or list all that apply to the ANS, or something like that. Just know it. Here are the answers. Enteric plexus, that's in the digestive tract wall. You don't need to know that here. We'll talk about that in anatomy and physiology too. But you do need to know the rest of it. So use this chart. Same as this. Here are the quick answers. But print this out and practice, practice, practice. Or draw it out and practice, practice, practice. But know this flow chart. All right, neuroanatomy. So let's look at the nerve cell itself. A nerve cell is called a neuron. Nerve cell and neuron are the same thing. They're synonymous. They're interchangeable. You're going to hear it called a neuron and a nerve cell. Just know that it is the same thing. It's just by preference. Sometimes I'm in and out of both. It just depends on what I'm talking about, but it's the same thing. Nerve cell, neuron is the same thing. Again, soma means body, right? Look how quickly that came up. So we're going to look at the cell body, aka the soma. So that's this right here. That is the cell body. It has a cell membrane just like any other cells. It has cytoplasm, it has Golgi bodies, it has endoplasmic reticulum, etc., etc., etc. All the same organelles are in a neuron that are in any other cell that we have. But a neuron is going to differ in a few ways. And how that neuron differs is what we're going to focus on. Let's see. So dendrites. Dendrites coming off of the soma. So these root things, tree branch things, those are little short projections off the body of the neuron, off the soma. Dendra refers to tree, and ite just refers to something being small. So these are like little tree roots, dendrites, or branches that receive information. Whatever you want to think of to make you remember that those are called dendrites and that they receive information. In the shadows here, you can see these little finger-like projections that we'll talk about in a second. That's this part, the axon terminal. So these little finger-like projections are sending information to these dendrites right here. So this is another neuron, another nerve cell, sending information to this nerve cell through the dendrites. The dendrites are picking up that information, sending it through to the soma, the body, and then down through the long tail called the axon. So in through the dendrites, out through the axon. Once we get to the end of the axon, like I just said, we have these little finger-like projections at the axon terminal. These little synapses. And at the end of each one, we have something called a synaptic bulb. But we'll talk more about what a synaptic bulb is. It's not labeled here, but you're going to see it here shortly. So remember that the end is a little bulbous, and we call it the synaptic bulb. And then this entire section is axon terminal, the end of the axon. The synaptic bulb is what's going to attach directly to a muscle or a gland or the next set of dendrites on another neuron. And that's what sends the signals right on to wherever the brain tells it to go. When we're looking at the actual neuron, you can see a whole lot of stuff going on, right? So first, let's look at the whole image. You'll see a lot of these little specks floating around. Those are called astrocytes, and we'll define what those are towards the end of the chapter. You'll see some bigger ones with a little halo ring around them. Those are called microglia. If we go back to the cartoon, oligodendrocyte, we'll talk about what that is here soon as well. But it makes a fatty wrap around parts of the axon. So what makes up the myelin sheath, which we'll talk about. Let's see. Node of Ranvier is just that little gap between the myelin sheath sections, but again, we'll talk about that shortly. Where was I? Perikaryon That is another name for the cell body or the soma. So it has three names: perikaryon, cell body, or soma. You'll hear me say cell body or soma. You're probably not going to hear me say perikaryon, but know that that's just a third name for the same thing. See, you can see the nucleus and the nucleolus. So nucleus there, nucleolus is the center. You can see the dendrites going into the cell body right there. More there. And the big axon coming off the cell body right there. The axon hillock is just where the cell body transitions to the axon. It's just a little transition area that we call the axon hillock. And I think that's it. Yeah, don't worry about the rough ER. I'm not going to make you label that, but yeah, know the rest of it, know how to label it. You can use this image to practice labeling some stuff. The astrocytes, the microglia, nucleus, dendrites, cell body, axon. If you're confused whether it's a dendrite or an axon is usually thicker than a dendrite. So see how they're these thin projections, these dendrites, thin branches, the axon's going to be a little bit thicker. So that's how you can differentiate between those. Astrocytes don't have a halo. Microglia do have a halo around them. So astrocyte, microglia, nucleolus, nucleus, the soma or the cell body, dendrites, axon hillock is that transition point. And what else? Oh, cytoplasm. Cytoplasm is what the fill-in of the cell body. I think that's it. Yep, so be able to label a nerve cell. Okay. Let's see. We just discussed oligodendrocytes and the fatty wrap around the axon. That wrap is called the myelin sheath. So here, the myelin sheath is that wrap around the axon. So pretend like that's wrapping all the way around the axon, and then we have little gaps. So wrap, gap, wrap, gap, wrap, gap, and so on and so forth. So wraps, leaves a gap, wraps and leaves a gap, all the way down the axon. So that only happens on fast neurons that need to send a signal very, very, very rapidly. And we call those neurons myelinated neurons. We also have non-myelinated axons, or unmyelinated. I'm not quite sure why that says non-myelinated, but unmyelinated axons. So obviously there's no myelin sheath in the non-myelinated axon, correct? Correct. So here there are chemicals inside and outside of the neuron, and they switch places all the way down the neuron, in and out, and in and out, and in and out, and over and over and over again. And this happens very, very slowly. They run every step of the way wherever they are. Myelinated, however, are fast. They just, like a passing play in football, they just pass right into the end zone. So what would happen when myelin starts to break down? What do you think would happen when myelin starts to break down and can't fire as rapidly? So think about that, and we will talk about it later in this chapter, and we'll talk about it more too in future chapters. So these are two different conduction pathways, right? Unmyelinated, we call continuous conduction. So it's continuously chemical in, out, in, out, in, out, in, out, slowly. Myelinated is called saltatory conduction. Salt, S-A-L-T, atory, A-T-O-R-Y, saltatory conduction. In saltatory conduction, you only have to switch chemicals where there's no myelin, which makes it much faster than continuous conduction. So unmyelinated, continuous conduction, myelinated, saltatory conduction. And then again, these gaps are called nodes of Ranvier. Not Ranvier, Ranvier. It's French. And again, just one last time, myelinated is fast, saltatory conduction, unmyelinated is slow, continuous conduction. Okay, so the synaptic bulb. So first we're going to talk about this image before we talk about everything that's written on this slide. Neuroanatomy, synapse, synaptic cleft. This is a synaptic bulb. So we are zooming right in on the synaptic bulb that's attaching to this dendrite, right? Synaptic bulb right there. So that's part of the axon terminal of this neuron. It's not part of technically this neuron, but it's about to send a signal, right? And so that's what we're going to talk about. So this is the synaptic bulb. So looking at it, you can see kind of a cytoskeleton in here with these like straight lines. You can see some mitochondria floating around, and you can see several bubbles. So the bubbles are vesicles. So remember what vesicles are in a cell? They hold things, right? They hold something. They're little packages. So these bubbles are actually neurotransmitter vesicles. So they are, in this image, doing something called exocytosis. So what's happening? One of these neurotransmitter vesicles, one of these bubbles, is coming to the edge of the synaptic bulb and it wants to merge. So bubble comes to the edge, and the outside of that bubble wants to merge with the synaptic bulb so that it can empty its contents, which the contents are neurotransmitters, outside. And then there are a lot of vesicles in different types of cells, and they do a lot of different things in different types of cells. But here in these synaptic bulbs, they are filled with neurotransmitters, which carry the signal from this neuron to this neuron, right? And we're attaching to this dendrite and wanting to communicate something over. Now remember, this is just an example of neuron to neuron, but it can carry a signal to a muscle or a gland as well. It can dump some neurotransmitters into muscles or glands. It doesn't have to be another neuron. But again, if we zoom out, we have one neuron, we have a soma, we have an axon terminal, and then we zoom into the synaptic bulb, which is this big image right here that we just talked about. Zoom back into the second neuron, and you can see where it's attaching to that dendrite. Again, the neurotransmitter is that chemical that's carrying the signal. And here it's carrying a signal across this gap to get to the next dendrite. The gap is actually called a synapse. So for a neurotransmitter to land somewhere, okay, what do we need? We have a neurotransmitter, it needs to land somewhere. We need something to receive it, right? Those are called receptors. So we just talked about chemoreceptors, pain receptors, temperature receptors. So the receptors are what receive information from the neurotransmitters. So we have the synapse, the gap, we have all these little receptors here. And these receptors, they have to be open and available to receive information, right? So if there's something blocking it being the receptor, if something is blocking the receptor, then the signal dies and it does not continue. The neurotransmitter just goes away. We'll talk about how it goes away. So it doesn't just go away into nothingness. There's a process that happens. We'll talk about it in the next slide. But yeah, so receptors have to be open and available to receive information, to receive those neurotransmitters. If they're blocked, then the signal doesn't continue. It doesn't get received. Sometimes we get prescribed medicine to block some of those neurotransmitters that are maybe overreacting or doing too much, or there's too many of them. Other times, disease will actually block receptors. So now let's talk about what we've got written here. So there's an amino acid called GABA. Gabapentin, that I'm sure you guys have heard of, is a drug that blocks that one. There are peptide neurotransmitters. Substance P is a pain signaler, so know that. Peptides are chains of amino acids, by the way. Substance P is just an example of a pain signaler. Endorphins and enkephalins are natural painkillers. So a lot of peptide neurotransmitters deal with pain, and a lot of enkephalins are actually working overtime in labor and delivery to block that pain. Anyways, if you ever talk to somebody who has been through labor and delivery, some of them will state that there was a lot of pain associated with childbirth, but the minute that they hold their baby, they're left with just a memory of that pain, that goes away. So those are actually the enkephalins blocking the pain signals. If we didn't have those, we probably wouldn't be as overpopulated as we are. If it weren't for these enkephalins blocking that pain, several women probably wouldn't want to endure that labor and delivery. I mean, well, now we do have epidurals and everything that we didn't used to have, but enkephalins are the painkillers that are involved that you think of when you think labor and delivery. Most of us have heard of endorphins. When an athlete gets their second win, that's the action of endorphins kicking in, basically. Purines, ATP, or adenosine actually inhibit the brain, so know that. Caffeine blocks the adenosine receptor, so it doesn't allow for the brain to be inhibited. So caffeine blocks those receptors, doesn't allow the brain to be inhibited, and that's why you associate caffeine with stimulating you or waking you up, but it doesn't actually stimulate anything. It just blocks the brain from being inhibited, right? So it just cancels that inhibition that you would feel. Gases and lipids, nitric oxide, NO, not no, just NO for nitric oxide, is made when it's needed, and it relaxes intestinal smooth muscle. All right, some of the other cells, neuroglia, also known as glial cells. We mentioned astrocytes earlier, but we didn't really talk about what they do. In the central nervous system, they're called astrocytes. In the peripheral nervous system, they're called satellite cells. And they're nearby regular neurons. So these cells maintain the extracellular environment. They get rid of extra neurotransmitters. So remember I said if the receptors are blocked, the neurotransmitters go away. So astrocytes and satellite cells are what that's what cleans that up. They direct neural growth, and then definitely know this one. Astrocytes only induce the blood-brain barrier in the central nervous system. Also, we know that's astrocytes only because it's in the central nervous system, and then blood-brain barrier, brain is central nervous system, right? So astrocytes are what induce the blood-brain barrier in the central nervous system. Blood-brain barrier is super important. Doesn't allow things to pass from the blood into the brain, which actually is super important in prevention of brain infections. Oligodendrocytes, they create myelin and makes an axon myelinated in the central nervous system. In the peripheral nervous system, it's called a Schwann cell, but it's basically the same. It makes myelin. So oligodendrocyte in the central nervous system, Schwann cell in the peripheral nervous system. So know that. Microglia, that's the astrocyte with the halo around it. They're the immune surveillance guys. So they're the security in the central nervous system, brain spinal cord, and they also perform phagocytosis. So if you remember phagocytosis from a previous chapter, those are like those little Pac-Man cells that eat or engulf the stuff that shouldn't be there. So they look for foreign invaders. If friend, do nothing. If bad guy, eat, essentially. Ependymal cells, that central nervous system only as well. They create and circulate cerebral spinal fluid, CSF. So that's the fluid that's around the brain, inside the brain, around the spinal cord, inside the spinal cord. If you've ever heard that someone had to have a shunt for too much water in the brain, it's actually that they had too much CSF, or the drainage for the CSF was blocked for some reason, causing it to build up in the brain. So that puts too much pressure on the brain or on the spinal cord if that's where we've got too much. And then a shunt is just a drain tube, gets inserted under the skull and allows for drainage and releases pressure on the brain. Here, this next slide is going to just show a table of what we just talked about. So you can read through that. Remember, astrocytes and satellite cells are the same. Oligodendrocytes and Schwann cells are the same. They perform the same function. They're just called something different in the CNS versus the PNS. This is just a short, informative section into some of the diseases of the myelin sheath, and we'll talk about this a lot later as well in an upcoming chapter. But multiple sclerosis, or MS, that's what happens when the myelin sheath starts to deteriorate, and it's going to disrupt nerve conduction. Onset's going to be between 20 and 40 years of age. It can be fatal from 25 to 30 years after diagnosis. Could be caused by autoimmune system, something related to the autoimmune, or it can be triggered by a virus. Vitamin D deficiency is associated with it. But this is when, that fatty sheath, the oligodendrocytes, the myelin sheaths in the central nervous system deteriorate. And then that myelin's actually replaced by hardened scar tissue, and it just causes a disruption, again, to that nerve conduction. Double vision, tremors, numbness, speech defects. Tay-Sachs disease, on the other hand, is hereditary. So here there's an enzyme that is lacked. There's an enzyme, a glycolipid GM2, in the myelin sheath. And normally it's decomposed by a lysosomal enzyme. Well, in individuals with Tay-Sachs, that enzyme is missing, and that accumulation of that glycolipid, let's fix that, disrupts nerve conduction. So too much. Unfortunately, this one is usually fatal before age 4, can cause blindness, loss of coordination, dementia. And now I just want to talk, while we're introducing the nervous system, I do want to touch on memory and synaptic plasticity, especially for, you guys that are taking classes and everything, so you kind of can get an introduction into memory anyway. So how memory works. So when we're learning something new, your neurons are going to form connections with each other, which is what's happening now as you're listening and taking notes on this lecture, right? So the more that you use those connections, the stronger that they're going to be, right? So, anyways, a memory trace is a synaptic pathway through the brain. So it's the physical basis for memory. So along this pathway, you're going to make new synapses, or the synapses that are already there are going to be modified or changed to make transmission easier, right? Synaptic plasticity is your brain's flexibility to adapt or change any connections. Synaptic potentiation is the process of making a transmission easier so it can make a connection stronger through use, or weaker through disuse, which we'll talk about. But a good example is when you're speaking a different language. If you don't use it, you lose it. As a kid, I lived in Germany. I was fluent in German, lived there, spoke the language. But now, because I'm never in a situation where I have to use that language again, I've lost most of it. If I were to immerse myself in the language again, it would come back. So the more you use the connections, the stronger that they become. And that strengthening of connections is something called long-term potentiation. Helps you remember. There are 3 different types of memory. There's immediate, short-term, and long-term memory. Immediate is what's happening right now for you. You're holding this information as it's being given to you for just a few seconds. Short-term memory is working memory that can last seconds to hours, like calling a phone number that you just looked at, or remembering that the dendrites go into the cell body and the axon is the tail that you just learned in the beginning of this lecture. That would be short-term memory. Long-term memory is something that you can remember up to a lifetime. And there are 2 different types. There's explicit, which we also call declarative. And these are memories that you have to think about to recall or to remember, like phone numbers that you memorized, for example, for your family members. At least my generation anyway. I know I've got a couple phone numbers that just live in my brain rent-free, right? That would be explicit memory example. Another one would be names of people or different important dates or birthdays. You have to think about them to remember them. And then we have implicit memories, so reflexive or unconscious memories. So these can be tied to emotional memories, like you got bitten or you got stung by a bee and it hurts. You got bitten by a mosquito and it itches. Those are those types of memories. You know that a mosquito bite's going to itch because you've been there before. And then you have procedural or motor skill memories. So these are things like riding a bike, driving a car, handwriting. All of those things are things that you will never forget. Which leads me to what happens when we do forget. So long-term potentiation is how memories are made. Long-term depression is when memories fade. So if we don't use a certain piece of information very often, the connections between the neurons are going to get weaker. Think of a sidewalk that gets overgrown with weeds. It's not really a sidewalk anymore. That weakening is called long-term depression. In this whole process, intracellular calcium is involved with memory. Low calcium levels are going to help the forgetting process. High calcium levels are going to help strengthen memory, right? But repeating information over and over again is going to help strengthen those neural connections. And then not using that information is what helps those connections fade. So all that's to say, study, make flashcards. Repetition is key so that you don't forget this information by the time it's quiz time or test time, right? Alzheimer's disease, we will talk more about. I did want to touch on it just so everybody's familiar with Alzheimer's, but we will talk about this in the next chapter. And then same as Parkinson's. We will talk about this in the next chapter as well. And that is it.